Take it for what it's worth. As you can see from the disclosure here, I didn't get anything from it, so hopefully you'll get something from it. It may not be worth anything, but we'll see. A lot of this comes from work that I've done with the CIBMTR. In that organization, we look at retrospective studies primarily, and we write protocols for every study that we do. And we found that these are very helpful in getting the studies done and in moving them along and refining the studies to get the best possible study that we can get. And of course, when we do prospective studies, we write protocols as well. So, well, what is a protocol? Protocol is a document that's prepared for a study that spells out what the objectives of the study are. It tells the investigators, other investigators, the public, the statisticians, what it is you hope to get out of the study. And as we'll see, we're going to want to write those in particular ways so we can make sure that when we do all this work of collecting all this data, that we can get something out of it. It spells out the design of the study, whether the design involves how we're going to select patients or what we're going to do to patients, or simply how we're going to go about collecting retrospective data and what we're going to collect and how we're going to look at it. It discusses methodology. Both medical methodology and statistical methods are going to be used to get the answers out. So, of course, it talks about statistical considerations, and then it kind of usually, particularly in prospective trials, gives an overview of who's responsible for what, so how the study is organized. And we'll go through each one of these in more detail. So, again, basically it instructs the study team on exactly what they're going to do, and this is perhaps the first and primary use of the study, of a protocol. All the protocols contain a brief, as we will see, section on scientific motivation for doing what the study involves. So that part of the protocol can be used to explain the study to the outside world. It can also be used to allow investigators to see whether it's worth doing the study by looking at the scientific basis of the study as it's written. And of course, if you're doing anything with human subjects, you need to get some kind of approval for the study, so the protocol is the basic document that we have to send to, for example, the IRB, or we would include with a grant proposal, or we would send to some agency to get approval to do what the study is in an appropriate manner. So what's in the protocol? Again, the key elements are the study objectives, the study design. We try to write these as testable hypotheses, the rationale for the study, endpoints, so a careful and usually somewhat technical description of what it is we want to measure so that we make sure that we're going to actually measure what we think we're measuring, and we can use that to test the hypothesis in an appropriate way. This may sound obvious, but particularly if you're doing retrospective studies, the way you measure things may well have changed from how you measure them today back to how they were measured 5 or 10 years ago. Or you may not have enough information to measure everything. So we want to very carefully, based on what the patient population is that we have, spell out how we're going to measure what it is we think is important. The protocol usually talks a little bit about safety reporting. This is something in a prospective trial, but not a retrospective trial. And of course, all of them talk about how we're going to, the statistical analysis, and who's responsible for what. Now, the standard outline that's used for designing a protocol has roughly 8 parts to it. And these 8 parts are sometimes included in the protocol, sometimes not. It depends on whether we have a prospective or a retrospective study. But almost all studies, this is the basis of what's there. So there could be things that were added. So we have objectives and specific aims, and I'll explain in a couple minutes the difference between those. Background and significance, so this is a literature review, why we're doing what we want to do. The plan for collecting patients and administering whatever it is we're going to administer to them, or how we're going to observe them. Again, a detailed description of what the outcomes are. Usually a detailed description of other variables that we want to include in and collect in the study. As we'll see, these are usually variables that you want to adjust for when you're doing the final analysis. And then statistics, concerns that are specific to the study that you're doing, a little hard to say, and a list of references. When we do protocols in the IBMTR, these things are fairly short. This whole thing is perhaps 3 to 4 pages for a retrospective study. So it's not a major outcome, major deal writing them, but it is important to write them. So the process that statisticians hope is going on, and hopefully you're doing when you want to do a study, is you go through this sort of checklist of things. You start out with some idea for something it is that you want to study. And this idea could be rather vague. Maybe you want to study cord blood transplants for leukemia. And then you take that sort of vague research question and you develop the concept, working with perhaps other people, perhaps with some statisticians, perhaps with some data managers, and the like. And you refine that concept into testable hypotheses. So things that can be readily translated from a sort of vague idea to a specific thing that we can test, and we kind of would know how we're going to test it using a particular statistical method. Of course, then you write the protocol, and there could be several revisions of that, revise it, finalize it, conduct a study, and then hopefully at the end of the study, you publish your results, and the study generates new questions that you can go and look at, and you kind of go through the process again. So this is the basic scientific method that's used to develop a study. So, as I said, we start with a scientific question. And we usually want to make these fairly simple and have 1 or 2 questions that are of primary interest in the study. We want a clear idea of what the study is going to answer. I don't know how many times I've had people come to me after they've collected the data and they don't quite know what it was that they wanted to ask. They just collected this data, and a lot of times when they come, they didn't collect the right data. So they want to ask some question, but there's no data there to answer it. So I want to start out with something that we're going to clearly write down as an objective. And usually we have 1 or 2 primary objectives, and then we possibly have several secondary objectives. And these are things that you can write out as sort of concepts. They're a little different than what we think of as the specific aim, which is rewriting these in terms of what it is we're going to measure and what we think about that measurement. So we may want to show that patients given a cord blood transplant have a 30% higher survival than patients that were given some other type of transplant. So that would be objective. An aim would be simply to show that cord blood transplants do better than others, or equivalent, or whatever. All right, then we take and we develop these ideas. So we need to then go to the literature, find out how to review in the literature how These general aims have been tested in the past. Perhaps what measurements were used, what's known about these things. Is there some historical controls that we may want to use instead of prospective controls and so forth? We think about generating measures of exposure and outcomes. So if we have a prospective trial here, we need to think about what the treatments are going to be when we're doing the concept development and how we're going to measure what the effects are. And a very difficult part of the concept development is picking the right control group. There's a lot of classic examples where very well-designed clinical trials were done, but they were done on the wrong groups. The famous example of a trial done at UCLA in the early days of chemotherapy where they had two kinds of chemotherapy, and one of them required getting treated every other day for a certain period of time, and the other one was like once a month. So they picked people that lived close to the hospital and they gave them the one type, and people that were far away, they gave the other type. And they found out, well, if you live farther away, you did a lot better on that type of chemotherapy than if you got the one for the close one. Well, obviously, there was a lot of bias in the control group here because you had to be healthy enough to come in and get the one treatment. And it was found that the exact opposite was right later on. So picking a control group is important. All right, the type of study you're going to do is going to modify what the protocol is going to be. As I said, there are two types of studies, or two ways of thinking about them. Retrospective versus prospective studies. One's where the data already exists, one where it's going to come into us. And observational versus experimental studies. Observational studies, we just observe. We don't require any treatment. We don't require any manipulation of the patient. Experimental studies, of course, does. So examples of retrospective studies are chart review studies, studies involving databases that are available. For example, the CIBMTR has a large database. There's a large database of solid organ transplants. There are databases for dialysis patients. There's a lot of big national databases from cooperative groups that you can use. And there are, of course, large governmental databases that make up prospective trials. So for a retrospective study, the protocol should focus on how you're going to pick data from this retrospective population of patients you have in such a way that they're going to be representative of your future patients. It's going to require a description of what the variables are, how they're defined, very careful definitions so that you can see that the definition you have now for these conditions in your current patients matches up with the historical definition. Usually when you do a retrospective study, the sample size that you're going to have is going to be fixed. You know in advance how many cases you're going to have. So what we then compute is the power of a test, if we're doing a test, how likely it is we're going to find a difference of a particular magnitude, or we fix the power, say 80%, and we compute what kind of a difference we can detect with the number of patients you have. And then based on these calculations, you can determine whether it's worth doing this or whether you need to go find more data. Usually retrospective protocols have tables in them when they come around of some descriptive statistics so that the major variables are already described. We do that to see if there's too many missing variables for one thing. And also, if we're comparing groups, we want to look at some of the demographics and make sure they're comparable in the two groups, so that the other factors other than what it is you're measuring are not going to affect what the outcome is. Prospective studies, the examples are our usual randomized phase 3 trials, phase 1 and 2 trials, cohort trials where we follow a group of patients over time, case control studies where we start out with perhaps disease patients, and then we go and we prospectively find healthy patients and match them up and see what's different about them. Now, in prospective studies, they could be, two kinds of studies. They could be studies where you make an intervention, or they could be studies where you simply observe. So prospective studies, you're allowed to sort of choose what you're going to measure. If you're looking at something and a particular biochemical marker is important for you, in a prospective study, you can write in the protocol at every patient, whether you're manipulating them or observing them, you measure that. If you go to a retrospective study, that marker may not have been measured, so you're kind of out of luck. So you can control what it is you're measuring in a prospective study. Okay. Now, as I said, the other kind of thing here is observational studies versus the experimental studies. Observational studies, you just observe. You make no attempt to modify behavior. And again, they can be retrospective or prospective. And as I said, they usually have simple protocols because you're not doing anything to the patient other than what the standard of care is or was. So these usually can be short, 4 or 5 pages. Experimental studies, the study dictates what the treatment is for each patient. So in your protocol, you need to write very carefully how you're going to treat each patient, what you're going to do if something happens to the patient that you're treating in terms of an adverse effect, what the mechanics of a drug would be, or the exact procedures and concomitant medications and things like that. So they're much more complicated to write because you got to write all this plan for what you're going to do to the patient. Experimental studies are also always prospective studies. It's hard to do a retrospective experimental study where you're changing things. So the protocol hopefully becomes a collaboration between not only you as clinicians, but between a statistician, a data manager, perhaps an epidemiologist, study coordinator if this is going to be a large prospective study, maybe a community representative if it's a large randomized trial. So a bunch of these different people, certainly a statistician, and here at the Medical College we provide free help in designing studies and putting together protocols. So we're available to help you with those kind of ideas. A lot of times writing the protocol is facilitated by having a template. So you have a list of an outline of what you need to do, and we'll give you one, and also at the end here give you a couple websites where you can get templates for various things. A lot of the times the templates, so they're available. They're available for different kinds of studies. And again, when you write the protocol, certainly what you're going to write is going to depend on the study. So you don't necessarily have to put everything in every protocol. So the advantage of having a template is that it organizes the essential information. It makes you write things that you may have forgotten otherwise. Kind of forces you to fill in all the blanks. It somewhat eliminates redundancy. You don't have to write the same thing in multiple sections, but it will show you where to write them. A lot of times when you get things that are reviewed, institutions, IRBs, I don't think we have this that much here, but sponsors, NIH, particular multi-center trials groups, all have very specific templates of the way you have to write things for them. Now, some disadvantages of using a template that I've seen is that quite often the template is like a previous grant that somebody wrote. You take that as a template and you have the, what you do is you just copy things directly from the old grant, the old protocol that don't apply to your grant. You get some of the strangest things when people are using a template that has sort of things as an example filled in, and a lot of people just copy stuff from those directly without realizing they don't match up what's in their study. The templates, of course, are generic. They don't apply to a specific study, so a lot of times you don't need to have everything there. So they're just too complicated for simple studies, and I think most of us at the medical college are doing fairly simple studies. We're not doing things. If you're doing a multi-center clinical trial, it's that complicated, you probably have 2 or 3 statisticians that are working on it and a whole bunch of people. So hopefully that's a little different. The other disadvantage is that the people work very hard to fill in the template and they don't really think about why they're doing it, and the science gets lost in doing it. So that's another problem. All right. So what goes in the protocol? So this is sort of the outline, and you see in some of these there's a star and some of the numbers, and the ones that have a star are ones that you necessarily wouldn't use if you were doing a retrospective study. They're just not needed. But every study needs a section, usually the first section, of objectives and specific aims. And objectives are usually clear and concise list of what the goals of the study are, usually written so that another clinician or layperson can understand what it is you're trying to get at. So they're not overly technical. They're technical enough to get through what it is you want to do, but they're a little bit generic in writing. They tell us what you want to study, what the questions are you want to answer, and who do you want to answer the question for. So even right away in the objectives here, we need to put a little thought in and thinking to who it is we're going to sample and who the final inference is going to be to. So, if we wanted to study some new weight loss procedure, we would want to say in the objectives, if we're going to only apply this in inner city folks, that this is a weight loss procedure for low income or whatever. We want to spell out, think about who we're sampling from, because the inference can only apply to the population that we're sampling from. Okay. Specific aims are a little bit different, and this is where working with the statistician comes in. And the specific aims, they're going to translate these objectives into some testable hypothesis. So if we were in the weight loss kind of thing, an objective may be to study a new, let's say, exercise program. A specific aim may be to show that people on this exercise program lose at least 10 pounds in the first month of the program. So we can take that hypothesis, take that and translate that directly into a test by taking their pre and post weight and testing whether that difference is bigger than 10. Okay, so specific aims are much more technical. They're usually given for both a primary hypothesis and a secondary hypothesis. And it's important to specify one primary hypothesis because at some point we have to do a sample size calculation, and that's usually based on the primary hypothesis. I know a lot of times I've gotten protocols, they want to know how big the sample size is, and they got 20 different things that somebody wants to test, but they don't tell you which is the most important. So to do that. The specific aims then, once we have them, should be discussed in the rationale, and they should be consistent with the rationale and consistent with the objectives. So the second part of every study is the background and the rationale in it. Here we briefly sketch the background for the study. We look at what knowledge is out there, give a survey of the literature, basically explain why it is we want to do this study. We also want to sketch the rationale for the study group that we're looking at. And if this is a prospective study with some manipulation, either drug manipulation or surgical manipulation or whatever, exercise manipulation, we want to sketch the rationale for selecting that kind of intervention and the level at which that intervention is done. That's what I mean here by the study dosage. If we're going to study a drug, we need to specify what level the drug is going to be tested at and why that's reasonable. Here we're really looking for the justification of the dose that we're looking at. Okay, as I said, we need to give a description of the device, the mechanism of action. So in the case of a prospective trial, why we're doing what we're doing. We want to also justify the population that we're going to be doing this in. And if we're doing, we also want a sentence or two about why we're going to study it in the particular design that we're going to end up using here, why we're using maybe a case control study, why we're doing a crossover study. If we were doing a study of some topical creams, for example, we may apply one to a person and then wait till whatever it was we were treating came back and then apply the other one. So that's a crossover design. So everybody's their own control. So why would we want to do that as opposed to not doing it? And why we can do it because we don't believe there's any interaction of things. So a little bit of why we're doing the statistical design that we're doing. All right, the third section in a protocol is usually patient selection. This is in like every kind of protocol. Now in a retrospective protocol, this could be relatively short, like, half a page. This could be, 4 or 5 pages if you were doing a prospective study, but we want to list how many subjects we want to enroll in the study. This is based on calculations we're going to do later. Indicate where we're going to get these patients from. If it's a retrospective study, whose charts they're coming from or what national database they're coming from. If it's a prospective study, whose patients they are, what clinic they're coming from. We want to discuss how we're going to evaluate these patients to confirm they're eligible. So if this is particularly something in a prospective trial, we may have certain blood tests or certain things that we need to measure or check on the patients to make sure that they're they're eligible for the test. Females, we have to make sure that they're not pregnant if we're going to be giving them something. So whether we want to just ask them or give them a test or something, you need to know. A lot of times this is done by making two lists here, listing what you need to do to be included in the study and then what it is that doesn't allow you to be on the study. So we have inclusion criteria and exclusion criteria, and usually just two lists of those. As I said, in the transplant registry studies, these are usually very short. 2 or 3 sentences for each one. We want to study AML first remission patients that were given some kind of myeloablative transplant or something. And so that could be the inclusion criteria. The exclusion criteria, maybe we don't want people that got whose donors were twins or sibling donors or something. We just want to do this in a particular type of donor. All right. Usually in the patient selection criteria, there's a little bit of discussion of the demographics of the available sample because we're allowed to look at the data as we're writing the protocol. Now, the protocol may require some refinement. We may add and subtract some patients, but at least for review and for discussion, it's helpful to have some tables there of numbers. All right, prospective trials. I'm gonna just kind of skim through this quickly. Prospective studies, we need to know what the drug schedule is, what the schedule is for actually measuring things on the patients. Okay, all the studies require some discussion of study design. So again, here is where we want to discuss very carefully how we're going to design the study. Is it going to be a cohort study, a case control study, a nested case control study, a 2-period crossover design? So you can work with the statistician and they'll help pick the best design so you get the most bang for your bucks here in terms of the number of patients you have and where the data is coming from. Prospective trials, we need to decide on how we're going to use placebos, how we're going to blind the patients or the clinician, or both. There's usually a little bit of discussion of how we're going to randomize patients in a phase 3 trial. How many study arms are going to be. We want a little discussion in the study design of how long it's going to take to get the data. In a prospective trial, this is important because sometimes it may take you so long that by the time you get the data, nobody cares anymore about what the question is. Things have moved on. So again, a prospective trial, sequence, duration of the trial period, who you're going to get here. A lot of times there are diagrams that explain the design that are given, especially if you have a complicated evaluation and administration of drugs. You have sort of a schematic that shows where people go. All right, every protocol has methods and assessments. So here, what we want is the variables that are in the hypotheses that we're going to look at. We want very careful definitions of what it means for a particular definition. For example, in the bone marrow transplant, there are a lot of different ways of defining relapse from leukemia. So we want a very careful definition. Are we talking about what type of relapse we're talking about? How is it measured? How do we define engraftment? What level of of platelets they have to get to engraft. Those things are defined for all types of studies. We also have to think a little bit about how we're going to deal with missing values. As much as you try, any study is going to have some people where you just simply don't know something for them. So we want to know, are we going to throw them away if there's not many of them? Are we going to impute the missing values? What are we going to do with them? So we want to do this for both the primary and secondary hypothesis, spell out definitions for all the important characteristics. We also want to provide a list of the other variables that we want to consider that are possibly going to be adjusted for. One thing that we like to do in the protocols, since this is also a game plan for the statistician, is to think a little bit about how we're going to treat variables. So if we're measuring age, do we want to treat age as a continuous variable, or do we want to treat it as adult versus pediatric, or over 40 versus under 40? Sometimes there are, suggestions in the literature that can be entered here and will allow us to work on things. All right, we also want to discuss here procedures that are going to be used. Again, discuss blinding. I took this last thing out from the NIH. They want to make sure that, in this case, we talk about how we're going to deal with pregnancy of females, specifically in a prospective study. In this section, for a prospective study, we'd also explain how often patients come, how often they're evaluated. For a prospective study, and this would not be in a retrospective study, we're going to talk about trial materials, so just a detailed list of what we're going to do with all the drugs and so forth. There's usually a section on treatment that explains the doses and how they're going to be given. How we're going to blind things, the concomitant medications and all that are here. Safety measures is a section that's suggested in all the protocols of what we're going to do for an adverse effect. Now, of course, this doesn't apply if you have a retrospective study. What is meant by an adverse effect, it's not always that easy to define an adverse effect. Sometimes death is an outcome and not an adverse effect. And who's going to deal with it? Who's it going to be reported to? Who's going to make sure that this wasn't caused by the treatment? It's a section on safety measurements. So again, how are we going to make sure the patients are treated appropriately, and who's going to deal with complaints? Now, all protocols should have a little bit about data management, where we discuss measures that how we're going to collect the data, how we're going to store the data. Are we going to store it on a file? Are we going to store it on a piece of paper, on a spreadsheet, in a database? If you want to store it in a database or a spreadsheet, we can help you work with that. Okay, and then a statistical section that deals with the size of the test and statistical methods. So we need to describe how we came about with the sample size that we have. So we're going to need to specify either the type 2 error or the power. Type 2 error is the probability that you declare there's a difference when there really is a difference of a certain magnitude. Or we can describe the detectable difference for a certain power. So here's a little table of kind of what we can do. If we have a retrospective study, usually the sample size is approximately fixed. It's fixed either by the total number that are available to us in the database, or it's fixed based on how much time and money we have to go back and review charts. Maybe we can only review 100 charts. So based on that, we can either fix some clinically important quantity that we want to detect, and we can compute how likely it is that we detect that difference. That would be the type 2 error. Or we could say, well, I want a type 2 error of like 80%. How big a difference do I have to be able to detect that? So the first one says, say I want to detect a weight change of 10 pounds. How likely am I to do that with the data that I have? Okay. Second one says, I want to be 80% sure that I'm going to detect a difference. How big a difference can I detect? So there are two different ways of looking at the same problem. And one or the other of those should go in a protocol. Usually prospective studies works the other way around. We compute the sample size, we fix a difference, and we fix an error rate, and based on that there's some formulas that'll give us the sample size. Okay, now some other studies, instead of looking at a test, you just want to estimate some quantity. And usually there what you do is you fix some precision, and you want to look at a sample size that gives you a particular standard error. That standard error, if you take 4 times that is, then you can be 95% confident that your estimate is somewhere in the interval of with 4 standard errors. Okay? So using that fact allows you to get a sample size. A lot of these standard errors depend upon the sample size. Well, they all do. And then, of course, the other thing that's in this section is the game plan for data analysis. That talks about the general considerations. If there's any safety analysis, like how many adverse effects can you see of the first so many patients before you have to stop the study, or how you're going to go about doing interim analysis, and what type 1 errors you should do the various interim analyses at. And then finally, you need something about ethical considerations. So these are basically the hoops you need to jump through to get things approved by the IRB. And can help you with that. Suggested that you have a policy for publicizing the findings. So who's going to write the papers and the like. NIH requires you have some plan for retaining the data after the study is over if you're doing an NIH study. So you need to discuss that. And then references. Some optional things are, particularly in prospective studies, are appendices that have the consent forms, the product use directives of the drugs and the like. Retrospective studies, again, usually some summary tables are there. Okay, just there's a few tips. They're sort of obvious here in writing these. Write out abbreviations, write version numbers, so you can keep track of these as you change them. Bulleted lists are good. put page numbers, short titles on each page. Again, the better you write it, the more likely it is to go through the various reviews that are out there. So, here's some references. There are a number of good clinical trials books. The one that I like is a book by Pocock. It's kind of an old book, 1983. Pocock is a nice book, particularly for clinicians, because there isn't a whole lot of, there's some statistics in it, but a lot of it is written at a relatively elementary level. But he has a lot on writing protocols and designing protocols, so it's a good book. I've used this book at other places where I've taught clinical trials courses for clinicians. And then I have 3 websites here. There's a government website that gives a template for writing protocols for prospective cancer trials, if you want to do that. There's another government site, another site that has a collection of templates, and I think there's 5 or 6 of them there, and they're for different things, like retrospective studies, minimal risk studies, randomized trials, phase 2 trials. So it's a good place to go and get a detailed outline. And then if you just want to see how we do it in CIBMTR, we have a website that discusses how we write simple protocols for the registry. So, as I said, if you want help, I think Kelly gave you a little flyer we have, so we're available for help. We have drop-in services 3 days a week here in this building, over in the TRU, which is in the Pavilion building. 2 days a week we have drop-in services in the Biostatistics department, which is over in the main campus building right underneath the library, so you can just walk in. If you're planning a study, you may be better off just going to our Biostatistics website and filling out an application. And we will then arrange a time that's convenient for you to meet with one of our faculty members and one of our master's level statisticians to help you with whatever it is you need to do. Services are free. We're not charging for anything. The CTSI grant now is covering all the things. All you need to do is check a little box that says it's translational research, even if it isn't. I shouldn't say that on tape, but so stop there. Any questions or comments or?